We are entering into next module, module 2, where I will be discussing about transcription and translation, particularly making a product from the stored information in DNA. During the last module, module 1, I have mostly concentrated on nucleic acids, particularly DNA, and we discussed about how scientists proved that DNA is our genetic material, followed by Watson and Crick's DNA structure actually help us to understand how information present in DNA and how it can be replicated. So that is very important step in biology. So we have to keep our information, particularly genetic information, safe in DNA, and it needs to be copied precisely without error for cell division, for next generation, for offspring, for everything. So now let's start with module 2, and today I will be discussing about gene expression and the central dogma of molecular biology. So here particularly I will focus on understanding the central dogma of molecular biology, followed by a brief introduction to gene expression, and also I will be discussing about the correlation between DNA content and its downstream product. So as I already told, DNA is nothing but just information. So DNA alone cannot work. So whatever information present in DNA, that should be translated, that should make some product which will be functional in our body. So those products I am meaning here, the downstream product, that could be protein. So now let's see here, flow of genetic information, which is also known as the central dogma of molecular biology. As I already mentioned before, that DNA goes to DNA, that is called replication, and then here mostly I will be discussing about the next two steps, that is transcription and translation in this module. And then as you can see that after transcription you will have RNA, which is also a polymer of nucleotides, but it's polymer of ribonucleotides. And finally, we will get protein after translation. So this is polymer of amino acid, and proteins actually do work for us. Now, in terms of discovery, I would like to mention here that DNA and protein, scientists understood much before the discovery of RNA, like DNA, how it's work, how it's replicate, and also scientists purified proteins, and people knew that protein, particularly enzymes, they have specific function, but RNA came little late. So later scientists understood that RNA actually connects the DNA and protein together. So RNA is the intermediate step. Without RNA, we will not have the protein. So from DNA, we have to make RNA, and finally from RNA, we will get protein molecule, which are functional in our body. Now, as I already told that in this module, we will mostly focus on this part, which is, I just put some box here, the transcription and translation, and different aspects of these two steps. Those are very fundamental steps in molecular biology, I would say in biology itself. And the term central dogma is is proposed by Francis Crick. So here, like I already mentioned that Francis Crick and James Watson, they received Nobel Prize for DNA structure, but also he proposed the idea of central dogma. So central dogma means here, generally we say this DNA goes to DNA, that is replication, then transcription, and then translation. That's all. But now, if we go into little bit in more details, we will get some updates in this flow of genetic information. So, as you can see here, that in 1970s, scientists, they were working with different RNA viruses. So, although I mentioned that DNA is our genetic material, but sometimes RNA can be genetic material of some viruses. And scientists which they were working with RNA virus, they found that they have some specific enzyme which is called reverse transcriptase. So from the name itself, it is understandable that this enzyme will make DNA from RNA. This is just a reverse reduction of the transcription. So RNA to DNA synthesis. And because of this enzyme, those viruses can reverse transcribe their RNA into DNA. And because of this work, David Baltimore and Martin Temin, Howard Martin, so they received Nobel Prize in 1975. This is very interesting work, and as many of you know that even HIV virus, for example, so HIV virus, they have RNA as their genetic material, and they do have reverse transcriptase also, and that helps in this reverse transcription. And also, if you see another group of scientists, they they were working with some RNA virus, particularly poliovirus and some other viruses. They found that those viruses, they are not sensitive to one drug. So this drug, name of this drug is actinomycin D. So this drug, it blocks transcription, particularly DNA-directed RNA synthesis. So if DNA is the template and when the RNA is being synthesized, this drug will block DNA-directed RNA synthesis. So now, when they found that these viruses, they are not sensitive to this drug, that means some other mechanism exists, that might be DNA to RNA is not, DNA to RNA synthesis is not happening. And they found that they have another enzyme that is called RNA-dependent RNA polymerase. So it can synthesize RNA out of RNA, RNA-dependent RNA polymerase. So as a result of that, RNA replication can also happen. So these are just some addition to whatever the normal thing I'm saying again and again, that DNA to DNA, that is replication, transcription, and translation. Now, because of this, many people, even many scientists, they were a little bit, skeptical about the idea of central dogma. That central dogma means dogmatic. That means it's kind of belief that which should be true. So that is the central belief of molecular biology. So that's why, people sometimes say that no, central dogma is wrong, some updates are there, something is happening here. But if you see, what Crick actually proposed, that is not exactly what we are telling here. So this is the, what Crick actually proposed. He proposed ideas on protein synthesis, and in a light-hearted manner, he actually termed this is central dogma. You can see whatever he mentioned here, this is very important. Once information has got into a protein, it cannot get out again. This is the major thing. This is the central dogma. So information means here amino acid residue. And see, generally in textbook we don't see this figure. But I would like to mention whatever this update I mentioned, that RNA to RNA synthesis or RNA to DNA synthesis, which were discovered later, much later, compared to this proposition made by Francis Crick. If you see, Francis Crick also mentioned all this possibility. Here, as you can see, that he mentioned that RNA to RNA synthesis, like here, that RNA replication can happen. Similarly, he also mentioned here that RNA can be converted into DNA, but never protein to DNA synthesis or protein to RNA synthesis and protein to protein synthesis. That is not possible. So, this is the central dogma, and in my understanding, whatever he mentioned that protein to protein, protein to RNA, protein to DNA, it shouldn't happen, and this is true also today. Like we cannot, like protein is not being formed from protein. So, in all this aspect, central dogma, whatever Crick proposed, this is like very much what we are saying now. Now, here like as I just discuss about the central dogma, particularly how the information flow from gene to functional molecule. But here like I want to discuss about like little bit different aspect. Here you can see three different plants. I named them here plant X, plant Y and plant Z. So, as you can see, all these three plants, they look different, but I want to mention here that all these three plants, they have same DNA because they are same plant, but they are looking different because of their habitat. That means these plants grow in land. So, this is terrestrial plant, and this is submerged. It is little bit inside water and outside water. So, this is submerged, and this is completely inside the water. So, that makes this huge difference, as you can see, that completely terrestrial, partially submerged and completely submerged. So, that means what I want to say here, the phenotype means the visible configuration of an organism, the phenotype, the visible configuration, what we see actually of this plant, this Mars plant, its scientific name Sagittaria sagittarifolia. So, it depends on its environment. So, although this is same plant, they have same like information in their DNA, but they look completely different. So, this is controlled by even by the environment. So, this is very interesting. That means environment can also regulate somehow the expression of gene, so that it looks different. Those three plants looks completely different. Now, in the same line I would like to mention this. Here you can see a skeleton of three different animals. So, here the first one is chicken, here this is chicken, and here this is rat, and here one snake or python particularly. So, here like one gene HOXC8. So, this gene is present in all these three organisms, and this HOXC8 is responsible for the formation of vertebra in the thorax region. So, as you can see, this gene is present in all these three organisms, and they are very closely related. If we see the sequence identity, that is also very close, because these three animals are not very far away in terms of their evolution, right. So, python, rat, chicken, but still their skeleton structure, particularly the number of vertebra, like differ a lot. As we can see, in case of Python, like everything is about vertebra and thorax, because this gene, how it is getting expressed during the early developmental stage, this gene is too active in case of Python, and it expresses a lot, and that determines the number of vertebra in thorax region. So as a result of that, what I want to say, although the information is present, but how this information is kind of getting like expressed, how we can read this information, because of that, the trait, the phenotype can be a lot of difference in between organisms. Now, like here, I want to mention about this, the correlation between DNA content and its downstream product. As I told that, like in this in that like slide where I was showing that 3 plants looks different, but they have same DNA. That means there should be some kind of correlation between DNA and its downstream product, and that's why the phenotype is different. So here, like Human Genome Project, it is a very one of the biggest project in biology, and it dramatically enhanced our understanding on gene expression. So this project, this Human Genome Project, was conceived, like I would say, was started in 1990. Like especially I would say United States of America, they have taken this initiative, but many other countries like Germany, France, UK, they have also joined in this collaborative project, and this is government funded project. And in 2003, they actually kind of completed this project, and we came to know the total information present in DNA of human. This is very important because of that, it revolutionized the biomedical field. So for the medicine, treatment, and genetic disease and many things. So it's completely revolutionized the field of biomedical science. But what I want to mention here, because of this Human Genome Project, we came to know that we have approximately 21,000 genes which are responsible to make different types of products. And it appeared to be significantly fewer than previous estimate. Like before that, we used to think about we have around 80,000, 1 lakh genes. But after this Human Genome Project, the number is very much low compared to the previous estimate. So it actually helped us to understand how this information processed and how many genes are present and their specific location in particular chromosome and all sort of information. And during this project, scientists, they have decided to sequence the whole genome of a set of organisms, not only human, because the human genome, the size of human genome, the amount of nucleotide present in whole human genome is really high. So better they should try with different organism also. We have to slowly progress, as you can see that it has been started in 1990 and in 2003 it has been completed. Full sequence was confirmed and mapped in individual chromosome or DNA in human genome. Now here, because of all this information coming out from Human Genome Project, here we can see like particularly I am showing here few organisms, like for example E. coli, which is a bacteria and it is prokaryotic cell, and their genome size. So genome size means the total amount of DNA in terms of nucleotide, in terms of base pair. How many, how much DNA is present. So this is the total number of base pair of nucleotides present in E. coli. And here this number is the protein coding genes, the number of protein coding genes. So what I want to mention here, that initially we used to think that like complex organism like human, like we have many cells. We have approximately 10 to the power 13 cells in our body in adult human approximately. And we have very complex system, complex organ like we have eye, ear, kidney, and so many complexities, brain. So that means we must have like many genes and many information, and that's why we are being like, we are very complex. So this is kind of general idea that complexity of an organism can be related to its genomic size also. But later on, it has been established, as I am showing here, you can see here, for example, this is E. coli, whatever I mentioned, this is yeast, some kind of fungus, and this is the genome size and the protein coding genes here. And interestingly here, this is C. elegans. So this is some kind of worm, very small worm, and this is the genome size, and this is number of protein coding genes present in C. elegans. So here I want to mention here C. elegans. It is very small worm. It has approximately around, I would say, 2000 to 3000 total cells in its body. Only 2000, something like that, in that range. But human, I told that like approximately 10 to the power 13 cells present here. And also, as you can see, C. elegans is very simple compared to human in terms of their architecture and organization, body plan, etc. But as you can see here, the number of protein coding genes are almost equivalent in C. elegans and human. Literally, I would say, in case of C. elegans, the number is little higher than human. But then why it is happening? So that means it suggests that the number of genes is not directly proportional to the complexity of an organism. That means in human, maybe the number of genes, the protein coding genes, are less compared to C. elegans. But the regulation, the crosstalk between the product of multiple genes are more complicated. And that is actually deciding that different types of function, different types of morphology, like organ development and all sort of things. So the regulation is like very important, the expression of gene and the regulation therefore. So that's why from here we would like to mention here that the number of genes or the just the content, how much nucleotides present, that is not directly dictating the configuration of a particular organism. So as you can see here, marble lungfish, this is one primitive fish. So if you see, the genome size is much bigger than the human or C. elegans, whatever you compare. But as you can see, this is not that evolved, right? So as a result of that, like how genes are getting expressed is very important. Like that is actually dictating many things in our life. So here, I would like to mention now the total DNA content versus transcribable content. Why? Because as I just mentioned that in that marble lungfish, although the DNA content is very high, but maybe like the products are not that many, or the regulation is not that complex. So that's why here we are trying to address this issue. So as you can see here, I just mentioned here that this is the genome size, in terms of base pair of nucleotides. And as you can see, this is E. coli and here human. Now, protein coding sequences in human is approximately 1.5%. That means although we have huge amount of nucleotides, like here as you can see 3.2 billion nucleotides present, 3.2 billion, but out of those only 1.5% of DNA can be transcribed into RNA, and finally which will be forming some protein, or which will help to make the protein, that's all. So now the protein coding sequence of DNA, so if I say this is one DNA, So double-stranded DNA. And here, if this portion is protein coding portion, so as a result of that, after transcription, it will make some RNA, right? After transcription, it will make some RNA. And that RNA is called messenger RNA because the message is present in that RNA to make the protein. So that's why it's called messenger RNA. So messenger RNA is the direct template for protein synthesis. So apart from messenger RNA, we have few more RNA which are also very important in making protein. So as you can see here that ribosomal RNA and transfer RNA. So ribosomal RNA, this constitute ribosome, and transfer RNA also participates in translation process. That means in helping in protein synthesis. I will discuss that in different class. So these are the three major group of RNA like messenger RNA, ribosomal RNA and transfer RNA. Apart from that, some other RNA also exist which we are not going to discuss in details. For example, microRNA, siRNA, all those things that helps in gene expression and gene regulation. But for our discussion, this should be fine, like mRNA, tRNA and rRNA. And now if just 1.5% of the human DNA that is being transcribed, so in that case most of the DNA sequences present in our genome are not transcribed. So that is kind of, they are just present but they are not getting transcribed. So here particularly I want to mention here something, because see, during every round of replication when cell divides, for example, that we have to copy all this information. Now during copying information, that means replication, like cells need to spend lot of energy and time, energy in terms of ATP or some kind of molecule that required for this processes. Then why we are actually maintaining that big chunk of DNA, although those DNA they are not directly making they are not getting transcribed and not making product. So actually I would say evolutionary it is very much helpful. So there are many reasons, there are many like I would say hypothesis, logic behind this. But very simply, if I want to mention something, for example, like throughout our life, for example, like human lifespan is, if I say around 80 years, 90 years, something like that. So during that time, we are exposed to different type of chemical, sometimes radiation for different reasons. But now, if we have only those precise thing in our genome, like a little bit of problem will show some defect in our body. But I want to mention here, just in this huge chunk of DNA, those important DNA are also getting hidden. So as a result of that, a little bit of change, like 1 or 2 mutations, that might not affect this crucial information present in DNA. And apart from that, we have different pathways where we can, in our body, in our cell, mutation or some little bit of damage in DNA can be rectified. But this is another reason I would say that will be helpful. For example, in case of bacteria, like most of their DNA is transcribable, not like human, just around 1.5% is being transcribed. In bacteria, it would be around 40%, 50% DNA can be directly transcribed. But those are unicellular organisms. If some big defect happens, the cell will die. It will not be a big problem. Before that, that cell already divided into many cells. So as a result of that, it is very important for eukaryotic complex organism like us, and with very long lifespan to maintain this information properly for many years. So this is all about this lecture, and that's all.